going forward with Borana Dollomaja from the Institut de Neurosciences des Systèmes. And she will talk about virtual epileptic patient cohort generation and evaluation. Thank you so much.
Thank you. Thank you for the opportunity to be here to talk to you about this topic. I'm suspecting that most of the audience is probably specialized in epilepsy, probably biologists or clinicians. I don't know how many people here are working computational models. I will try to convey basically what we do and explain to you the utility of what we do. Thank you. Here I'm focusing also, just like the previous speaker, in drug-resistant epilepsy, affecting 1 in 3 people with epilepsy, with brain surgery being the main treatment option, as this was previously said also. And currently, especially in France, but more and more now also in international centers, it is routine to implant intracranial EEG electrodes, like was shown before, in order to go in deep structures of the brain to measure electrical brain activity. Here I'm showing also a recording of a seizure event for a patient. And from this, clinicians have the expertise to then estimate regions if the epilepsy is focal, to estimate where the seizure comes from, make a surgical decision, and then the patient can basically go into brain surgery, resective surgery. However, the success rate, even though it varies from one type of epilepsy to another, is about 50%. So this is not, it could be better, basically. It's something that we could improve. It is thought the failure of a patient being seizure-free after surgery, it's thought to be due to either a partial diagnosis of the epileptogenic zone or a misdiagnosis of it. So basically, diagnosing the epileptogenic zone network is important, and we need to find ways to improve that. And this is where I'd like to show you how people who are not necessarily clinicians, but people who do brain modeling, computational brain modeling, could potentially help, and how we can do that through whole brain models. And then talk to you a little bit about how we can also build synthetic data sets from that and how that can also contribute. So what are these whole brain models for epilepsy? So when we talk about modeling, what we first like to do is we like to look at patterns in data, and we like to look at specific things that could be interesting or important to measure and to model. So in particular, something that I find very interesting is that seizures across species have some universal patterns. So these are local field potential recordings from different species, the mouse, zebrafish, and human. And all these seizure recordings have some particular similar properties in terms of their dynamics. They have interictal spiking in them. They have fast discharges. As the seizure goes, as the seizure progresses to its end, there is a slowing down in the signal. So we could model this basically, and the way we could do this is by using differential equations that allow us to model dynamics, allow us to model how something evolves in time, essentially. And here what we want to model is how electrical brain activity evolves in time. And in particular, we want to model these fast discharges and spike and wave events, interictal spikes, and we do this through what is called also neural mass models. So they model the activity of a population of neurons. What is interesting also, in this particular model, we have a parameter, x0, that is the excitability parameter, we call it, and it allows to set a system to either be in a seizure state or in a non-seizing state. And this is guided through this variable z that evolves slowly in time, that is inspired also from real recordings where we don't only see fast discharges, but if we measure also oxygen or ATP consumption or extracellular potassium, these seem to be correlated with also seizure events. Now, this can be interesting in particular if we also take into account patient-specific brain imaging data. So for one specific patient, we can model their brain, of course in a simplified way, by taking their T1 scan parcellating their brain using atlases, representing brain regions as nodes, and then connecting these nodes to each other through tractography. So the white matter fibers will help us then estimate the connection weights between the different regions. And then what we can do that we can put in every single node, we can describe the electrical brain activity of every single node using a thing such as the epileptor model that I showed you before. And then what's even more interesting is that we can couple these models to each other. So the brain activity of one node will then influence the activity of the nodes connected to it and vice versa. So then this can give rise to complex dynamics. And from this then we can, we do simulations on the whole brain level, but we can map the activity to the reconstructed SEEG electrodes. And so here I'm showing an example of such a thing. So simulated spontaneous seizures that use an epileptogenic zone hypothesis from the spontaneous seizure. So we use that to parameterize the model, especially the excitability of every node. And this can be interesting also as an approach to also automatically estimate the epileptogenic zone. So the way this is done is we take the same virtual brain model we built, which is personalized for every patient, it's unique for every patient. And essentially, we can do data fitting, in particular via methods such as Bayesian inference, where we can run simulations by changing systematically the parameters, the excitability parameter of every node in our model, for example, and comparing that to the features of the real SEEG recordings. And the best match will then give us a distribution of excitability. And so what you see then as an output, we can have a list of regions with their different excitabilities and an epileptogenicity heat map. And this is what we currently do in a clinical trial in France. It's still ongoing, but it's soon coming to its end. And hopefully this can be a useful tool to the clinician to improve the success rate of resective brain surgery. And as a second step, so I spoke about virtual brain models. We can run simulations. So something we can also do quite easily now that we have all of this set in place is that we can create virtual cohorts. So based on based on real empirical data, we can build personalized brain models for every patient, and then based on their patient-specific epileptogenic zone hypothesis, we can build then cohorts of synthetic versions of their SEEG recordings, so spontaneous seizures, but also stimulation-induced seizures. So these are seizures that are induced by stimulation. I will go a bit more into it, and also interictal activity. So we can do this with the epileptor model by basically tuning the parameters for each patient such that we can then obtain patient-specific synthetic data. And this is a synthetic data set that is hopefully will be useful to people who are also doing data analysis methods because the advantage of synthetic data is that not only they're easy to share and anonymized completely, but they also have the particularity of having the excitability parameter that we set up in order to get those simulations. So we also tried to estimate how well the match between our simulated and empirical recordings was, and for this we needed, basically we wanted to look in particular at the spatiotemporal dynamics of seizures at the level of the electrodes, and not particularly at the complex frequency or amplitude patterns, because the epileptor only captures so much in the signal. But in terms of spatiotemporal dynamics, if we binarize the seizure recordings, we can then compare the binary images to each other. So we can use correlation methods or overlap methods, basically as simple as we can get. Then in order to compare how well this matches, we can build surrogate data where we take random epileptogenic zones from other patients, compare the same simulations, the ones coming from random EZs, and we see that basically the performance is much lower in red shown here. So this means that a personalized epileptogenic zone hypothesis is important in capturing features from the empirical SEEG. And we can do the same from stimulated seizures. So in this case, the epileptogenic zone network estimation comes from the spontaneous seizures of the patient, but we can run simulations of stimulated seizures. We can do the same comparison, and it turns out that even though we use the epileptogenic zone hypothesis from spontaneous seizures, we still can capture dynamics of stimulation-induced seizures. So that's quite interesting. Just to go quickly, so now this is an avenue that goes a bit beyond. In particular, in brain stimulation, we are very interested in that. The stimulation-induced seizures are not so easy to get clinically. Clinicians have to go into many trial and error procedures where they will stimulate in different places with different parameters of amplitude, frequency, and so on, until they can get... Sorry, this is my timer. So I will end here, but just to end in this last slide. Basically, it will take them a long time until they can get to stimulate in the right area. And they do this with the goal of better delineating the epileptogenic zone. And our goal here is to use these whole brain models that I just showed you to see if we can improve or guide them, at least in the search for these optimal parameters, so that they can have a more informative view of the epileptogenic zone. And just one simple thing we did here, if we grouped the stimulations that induced seizures, so this is just one patient, and the stimulations that did not induce seizures, it seems that the ones that did were much more focal in their electric field estimations than the ones who did not induce seizures, who were basically applied all over the brain. And it seems that this stimulations that induce seizures matched to the estimated epileptogenic zone. So it seems that there's something that, by modeling approaches we can do. We can maybe put some more insight and hopefully have some more be of help basically to patients and also to clinicians. So I will end here. So these are just extra slides where I go into looking at different stimulation parameters, amplitude and distance, basically just running different simulations and comparing the results. This is still preliminary. There's a lot more to do, but just we see that just for now, it seems promising. It seems that these models can give some insight. In particular, these virtual cohorts, their advantage is that they really personalized for every patient. They have a patient-specific epileptogenic zone, and that seems to help in capturing these empirical features that we observe. So I hope that also the virtual cohort can be of help to the entire community. So if you are interested, the data is available, also the code that you can run your own data with. In the future, just to end on that note, here we're not talking about mechanisms yet, but mechanisms are important. We also are curious about that. For that, we need different models. We need biophysical models where stimulation parameters can have a direct link with biology. So we also need to think of using better models, basically, in the future. And with that, I thank you so much for your attention. These are the people from my lab. It's my supervisor's birthday today, so I put also a picture of a cake. Thank you so much.
If there's one burning question, otherwise we move forward to